During today's webinar, my colleague Lisa, who is also an application specialist, will tell you why you should label your tubes, but not your cells. And if you have any questions during the webinar, please do not hesitate to ask them in the chat box. We will answer them at the end of the webinar.
All right. Hello and welcome everyone, and thank you for joining in. I thought it would be actually nice to warm up a little and start this webinar with a little quiz about cell imaging techniques. So have a look here. Do you recognize all the different techniques the cells were imaged with? I will give you a couple of seconds to have a look. I guess let's have a little poll maybe. Are you an imaging expert already? Just click by answering. Let me just wait a couple more seconds. Okay. So for those of you who thrive to become experts, let's reveal what lies behind those images. I will start from the top left. This detailed image is from a transmission electron microscope. It's used to study the internal structures of cells down to an atomic level. Next to it, we have scanning electron microscopy, also using an electron beam to scan the sample and get information about the surface and the composition of the sample. Next one up is atomic force microscopy. You might have seen this one before. It is also a surface scanning technique that has sub-nanometer scale resolution. And bright field microscopy is probably your friend in the lab if you're working in cell cultures. Next one, you might have seen it before, is a phase contrast microscope. You're able to see the cells because they're causing a light phase shift, which is actually something I'm going to talk about later a little bit more. And now, this is my favorite because that's also something Laura and I are working with every day. This is digital holographic microscopy. With digital holographic microscopy, you're able to not only see your cells, but also quantify them for the different cellular parameters. For example, you can do that with our HoloMonitor system. So this is going to be exciting. This one is a bit tricky, but I guessed you probably get it right. This is the little monster microscopy. Oh, what a funny face. The next one is fluorescence microscopy. Those colorful images are made that the cells or the inner structures are labeled with fluorescence markers, and then you can see them. A special type of fluorescence microscopy is confocal, where the fluorescence light beam is focused to a small part of the sample to reach super high resolution. And speaking of that, storm, palm, and STED are also high resolution techniques. To get a little bit more order into this, I highlighted for you now in green the imaging techniques I personally feel like are live cell imaging techniques. I, for example, however, dotted the palm super resolution because of the phototoxicity problem you face when using high power light sources. Also now highlighted in red are the imaging techniques that don't require any cellular markers. You work label-free, which brings me to the central question of this webinar. Why should you go label-free? This image, or this video here, shows you a label-free time-lapse imaged with our HoloMonitor system. And you can see the cells moving in to close a gap. This is a wound healing assay, and it is label-free. So let's have a look. Why should you go label-free? First of all, it is cell-friendly. It is a non-invasive method with absolutely no need for toxic chemicals that destroy your cells. So if you can work label-free, just do it. In label-free imaging, your cells remain in their natural state. So there's no change in cell behavior or its integrity because of the imaging method, which allows you then also to do biological relevant long-term studies. As in label-free imaging, you less manipulate your cells. You can also trust your results more, right? So with this, I also want to just paint a little picture for you. Imagine you have a house. Hopefully very much decorated for the holidays, but that's not the point. Imagine this house and its inhabitants going about their daily life. And then suddenly you make, for example, holes in the roof and in the walls for you from the outside to be able to look inside. Now imagine as well it's snowing, so that all the snow and the stuff from the outside would just drip into that house through the holes. Do you think that the inhabitants would still react normally and go about their holiday preparations? No. That's what you do when you label cells. You cannot expect them to keep their natural behavior. Label-free live cell imaging doesn't need any extra steps in the protocol. It requires less work. You don't need any special media or additives. And it's great, therefore, also from an economic point of view. You would spend less time and fewer steps to get your data. And as we at PHI here are based in Sweden, we love the environment. And with label-free live cell imaging, you can reuse your sample. For further experiments, you can get therefore more data and more comparable data from the same sample. So that's great, because all our cells are precious, and working, for example, with primary cell cultures or stem cells, saving your sample is crucial. So we promised you to explain a little bit in the next part the technology of digital holographic imaging, and which is a label-free technology. But before I want to jump into that part, let me ask you one question. Have you ever seen a hologram? Just again, click the answer. Okay, this is going to be exciting, actually, for the most of you. Thank you for answering. So, what is a hologram? Depending on whom you ask, it's actually not a picture. It is, which sounds maybe a little bit odd, a photographic recording of a light field. So have a look at this little bit grisly image here. That's a pattern on a photo paper that, if you would shine the right kind of light on it, will reflect the light rays so they will bounce back, form an image. They will form a hologram. A holographic image can then be viewed from different angles as if the object is really there. It's like 3D-like. You can see the objects without the help of any special glasses or other optics. And that makes it, for example, possible for when you have a look at this picture, that's a German circus, which is using holography in their show. To me, that elephant looks pretty real. So, how's a hologram created? Let's think of light as a light wave. Let's do that first. And then let's go back here. So we have here in the first picture a coherent light beam coming in from the left. It will be at the beam splitter divided into two. One light beam will be mirrored and remains unchanged. That is the reference beam. It has the same properties as the original light source. The second beam will go through the object and will be changed by the object in its path. And then when both light beams meet, they will create what is called an interference pattern. That is what's captured on the photographic plate. That's that stripy pattern. That's the interference pattern of the two beams. You can think of that pattern a little bit like the pattern the leaves and the drops are creating in the waters when the waves are crossing over. So the holographic image is then created by illuminating the pattern with the laser light that was used before. It creates then the same light wave field that was originally coming from the imaged object. And now you can see the object again itself. Super cool. A traditional hologram is recorded on a photographic plate, but with the development of computers and high-resolution image sensors, it has been made possible to instead record a hologram digitally, allowing the holographic image to be created by a computer. But how do we see cells then? There is an issue with live cells, as living mammalian cells are actually as see-through as ice cubes in water. So how do we see them without actually any stains, markers, or labels? So let's think of cells also as objects where the light must pass through. Light waves of a specific wavelength have two principal characteristics, which is amplitude and phase. Amplitude corresponds to light intensity and is the height of a wave, and phase describes whether the wave is currently at its top or at its bottom or somewhere in between. When light then passes through a cell submerged in cell media, the more optically dense cell slows down and delays the light slightly relative to the surrounding light just passing through the media. And that is causing a phase shift of the light. By using a phase contrast microscope, you can visualize and observe those phase shifts and make therefore unstained cells clearly visible. However, conventional phase contrast microscopes, they cannot quantify those phase shifts, only visualize them. And that's where quantitative phase imaging comes into play. By using a digital image sensor and sophisticated computer algorithms, you can now have the ability to both quantify and visualize those phase shifts. And that's how our label-free live-cell imaging system, the HoloMonitor, works. In the next part of this webinar, I want to introduce you a little bit more to our HoloMonitor system. You have a schematic drawing here. So instead of light hitting a photographic plate and giving the interference pattern, the combined light beams after passing through a sample, for example your cells, will hit a digital image sensor. The hologram will then go into a computer, which then will be able to reconstruct the cell image. Because everything is calculated, everything can be given back to you as hard numbers. So it's a quantitative method as well as qualitative. So you, for example, you're able to see how thick a cell is in the image, but you can also calculate the thickness of it. So here we have two examples from our HoloMonitor, how these reconstructed images are looking like. On the left-hand side, flat from above, you have the 2D view of your cells. And because it's a holographic image, you also have the 3D qualities. You can see your cells from every angle. The coloring, by the way, is a pseudo coloring which is made afterwards in the software. And it's a way to show how thick a cell is. So if you have a look at the color bar at the left-hand side of each image, it indicates that a thinner part of the cell is more like a yellowish, and thicker parts of the cell are shown in purple. And that makes it also possible for you to distinguish different cell thicknesses in a 2D image. That looks all very pretty, but what can you do with it? So the first step to do when you work with a holographic image is to identify what is a cell and what is a background. So let's have a look at the left-hand side at the conventional phase contrast image. You see there's a blue line across about, how much is that, like 5 cells? And you have a profile underneath showing the intensity of the image. It is not easy to distinguish what is cell and what is background. And also you have these white hollows around the cells. If you, on the other hand, have a look at the holographic image, there's the same blue line crossed about like the same 5 cells. The profile of the cells is much clearer. It's obvious where there's a cell and where it's background. And that makes it also easier for computer software to identify those cells. In our HoloMonitor App Suite software, the cells are segmented and identified, and therefore then circled and marked. And as soon as you do that, you can create hard data, and you get like the information from that particular cell already, just by identifying it. You can get information, for example, on a visual cell level, on its area, volume, thickness, how's the texture of the cells, and how irregular is the outline of it. And that's a benefit from holography. You can get hard data on your cells. From a single cell, we can have a look now at the whole image. So here's a screenshot of our App Suite software and the morphology application, where again in the picture we see the cells were identified and circled here with a blue color. And because that happens, you can now also get data on the cell population. On the left-hand side, we see that these cells in the picture are now compared for the optical thickness versus the optical volume. So literally just by having the image, you can get morphology data of your cells. You see how many you have and what is the confluence of your cell culture. And that also makes it possible for you, just by having this image, to test and study different, for example, like proliferation, differentiation, or cytotoxicity of your cell. Oh, what is that thing doing? Or for example, have a look at your cell quality. So far, we only had a look at one image at a time, but because you can place the HoloMonitor system actually inside in the incubator, you're able to conduct time-lapse studies and follow your cells over time. Like here, this is a 24-hour label-free live cell imaging video of melanoma cells. And you're able to follow the single cells. You see them divide, right? You can see them move around. You see how they interact and how they shift shape over time. And you can see all that. Let's just continue watching this a bit more. I'm always very fascinated with that. So these were the images. Now what you can do with it. You can use HoloMonitor for a variety of cell biology applications. As I mentioned before, you can check your cell qualities and get information on cell numbers and confluence. And because you can put our HoloMonitor inside the incubator, you can conduct kinetic studies using time-lapse images. For example, checking different treatment effects on your cells. How are your cells moving? How are they growing? Or for example, how are they closing a gap in a scratch or wound healing assay? In the in-depth analysis of our software, you are also able to track individual cells and study cell morphology changes over time. And again, that's all possible from a single experiment setup with our HoloMonitor. So I thought it would be nice maybe to present some recent publications from this year, which are featuring HoloMonitor. So Makdasi and colleagues used HoloMonitor to test treatment effects on HeLa cells. They used time-lapse images to actually see the effect, but they were also able to quantify some morphology parameters over time, like we see here, area, irregularity, or roughness of the cells. Another example here is from the field of stem cell research. Owston and colleagues used HoloMonitor to compare different cell characteristics. They were able to visualize, but also quantify the morphology of dividing and non-dividing stem cells. And also they were able to observe migration patterns and study different cell motility characteristics. I've got a third example for you. Here the HoloMonitor was used in a wound healing assay. Varon and colleagues studied cell movements, and they could identify characteristic migration patterns for different cell treatments. And they also used the time-lapse imaging to assess the influence of long non-coding RNA on cell motility. Oh, all right. Because this question just came up, yes, you can find all our publications on our website under the, the publication tab, right? Yeah. So just have a look there. Yeah, that brings me already to the final slide, guys, with the take-away or the take-home messages, which are the reasons why you should go label-free. Label-free is cell-friendly. It's a non-invasive method. Oh yeah. And cells are able then, therefore, to keep their natural state and behavior for you actually to get more trustworthy data. And also, we, with our HoloMonitor, are thriving to make actually label-free live cell imaging even easier. You're able, with, for example, the HoloMonitor, to conduct different cell biology studies, ranging from kinetic studies of cell movements, tracking your cells, having a look, how is their morphology, how do they differentiate. It reaches also to actually testing drug responses or having a look at cytotoxicity. And don't forget that you can get individual cell data as well as population analysis. And with that, I thank you for listening to our first PHI webinar and hand back to my colleague Laura. Thank you.
Thank you, Lisa, for the very exciting talk, and thank you everyone who has listened. So now we're actually going to answer some of your questions that we got during this webinar. So first up, we have Patricia, who is actually interested what is the resolution of the system and how much is it possible to see. So Lisa, if you don't mind, I'm going to answer it.
Yeah, go for it.
So thank you for the great question. HoloMonitor has a lateral resolution of 1 micrometer. And I guess by how much you can see, you're wondering about the field of view. which is about 500 by 500 micrometers. So you would get information on your individual cells and cell populations at the same time. Let's see what another questions we have. So we have a question from Fred, who is interested what type of cells can be imaged with HoloMonitor.
Yeah, I can answer that. Hi, Fred. So with HoloMonitor, you're able to image all types of adherent cells, or for example, cell lines. That reaches from, also from primary cell cultures to like stem cells, macrophages, or for example, cell lines like HeLa. But if you have any questions, like everyone, you're always welcome to drop us an email. Do we have any more?
We have one more question from Eric. He's working with stem cells and studies their differentiation. And out of one of our publications, he's interested how he would be able to set up this experiment. For example, if he wants to study cell changes and motility changes, cell morphology and motility changes. So that's a wonderful question, actually, Eric. So you can do it with a HoloMonitor with one experiment inside your incubator. We would suggest you to set up a cell kinetic experiment where you could reuse that data to get your cell morphology data. And we will get back to you with more information on that via the email. Thank you everyone for your questions. However, we are running out of time, but don't worry, we're going to answer all your questions via the email. If you want more information, please visit our website or write us an email. If you want to get updates on our future webinars, events, or exhibitions, follow us on social media. Thank you very much for your time and have a wonderful day. Bye.
Bye.